and so here's BCI Applications within AI. I'm Ayushi.
I'm Ron.
I'm Maya. So BCI is a brain-computer interface, and it's a form of direct communication between electrical activity in the brain and a computer. And BCIs can connect the human brain with external devices. An example is a prosthetic, like you can see on the screen.
Yeah, so how do BCIs actually work? First off, the user has a mental task that they think of. It could be like an action, a movement, or a word. And then that signal is acquired via electrodes on the brain, and we'll talk about that coming up. And then the signal is then processed, the noise is reduced, and we actually isolate the parts that we're actually interested in, and the features are extracted and decoded. And then we move on to this device interface part, which connects the user's thinking with something in the physical world. So it's the command is sent to an external system. And lastly, we have feedback, which is the sensory information returned to the brain so that they know, they get feedback on the task that they've completed. Yeah, so moving on to the technical content. So how do you actually acquire neural signals? So there's three main ways that we'll focus on today. The first is EEG, which are scalp electrodes. So these are the non-invasive measures. Also, because it's non-invasive, it has a lower spatial resolution, which means the noise is highly sensitive to, the data is highly sensitive to other kinds of noise like muscle twitches or eye movements, things of that nature. And the next signal acquisition method is ECOG, which does require surgery, but it's also less invasive than our last method we'll talk about. It has a higher spatial resolution as it's closer to the neurons that are firing. And lastly, we'll talk about deep brain and microelectrode implants, which are a fully invasive measure. They're implanted deep into the neural tissue with the highest level of precision and most stable neural recordings. Okay, so moving on to signal cleaning and feature preparation. First off, cleaning is necessary because raw brain signals are already weak on their own and they're mixed with noise. So we want to isolate the parts that we actually care about, and we improve the reliability before moving on to the decoding process. So the signal cleaning method we use is independent component analysis, or ICA. With that, the problem that it kind of solves is that recorded signals are a mixture of brain activities and other artifacts like we talked about. So what it does, it separates these mixed signals into independent sources and removes the non-neural components that aren't necessary. And so these are the most commonly applied in EEG-based BCIs. And you kind of think of it as an unmixing of overlapped voices in a crowded room. And then the feature preparation process uses a method called common spatial patterns, or CSP. So the problem that this solves is that different mental tasks will activate overlapping brain regions. So what it does is, instead of removing the noise, it finds spatial patterns that best distinguish between two different tasks and enhances the differences in these signals to help understand which task that the user is actually trying to do. So you can think of it as like two people with microphones in a stadium, and CSP finds the best way to combine microphones so that one combination mainly captures person A, whereas another combination mainly captures person B's speech. And then moving on to linear discriminant analysis for neural decoding. So this is the heavy chunk of the decoding process. It is a supervised classification algorithm that finds the best linear boundary that separates different brain activity patterns. So it's used to classify mental tasks, for example, moving your left hand versus your right hand, like motor imagery, movement versus your rest position, or you can use it to select letters in a brain-controlled spelling system. So it finds a projection of the data, like you see on the left here, that maximizes the separation between two different classes or multiple classes, and it minimizes the variation within each class. So this makes the neural signals easier to distinguish, to understand what the user's intent was. And it's also the most common machine learning model used in brain-computer interfaces. So it's fast enough for real-time decoding, and it works well with small neuroscience datasets, which are most common in this field. It also pairs really well with the CSP features that I talked about earlier. Cool. And moving on to the last slide for me, it's device interface and interaction paradigms. So device interfaces are things like prosthetics, cursors, wheelchairs, and speech systems, which the user actually interacts with using the brain-computer interfaces. So these decoded signals become ways to control these device interfaces. And that, moving on to interaction paradigms, there's two different types, discrete commands and continuous commands. So a discrete command would be a single click or selecting a letter, and a continuous command would be moving a cursor in 2 dimensions. So the designs need to balance accuracy, speed, and effort. So if you use a different model than the one I talked about earlier, which could be, for example, stronger, it might also take more time to decode these to do the neural decoding, which might make it less viable for continuous command, for example, because of the latency involved. Moving into feedback mechanism, this is the last step to close the loop in the cycle. It's usually a visual, tactile, or auditory kind of feedback, and the goal is to reduce the training time and error so the user learns faster how to use these BCIs, and the feedback closes the loop and also drives adaptation and iteration throughout the model. Yeah.
So now for the clinical content, some clinical applications of BCIs are for helping with motor disorders such as paralysis, spinal cord injuries, or Parkinson's disease. And one way they can help is by translating neural activity to commands to allow users to connect to prosthetics, or they can also aid with physical therapy. And combined with deep brain stimulation, which is like when electrodes are planted in your brain to stimulate different parts of the brain, they can help modulate neural activity. And BCIs can also be applied for communication disorders, such as working as communication aids for aphasia patients, and they can promote neuroplasticity and help with recovery. And they can also make synthesized speech based on someone's neural activity. Some more applications are for cognitive and mental disorders, such as Alzheimer's, dementia, and other disorders that impact processes like memory, problem-solving, attention, and language. They can aid in cognitive rehabilitation by interpreting brain signals, and they can also promote neuroplasticity and can help with the reorganization of damaged pathways. And they can also facilitate cognitive assessments, such as diagnosing for different disorders. And also for sensory disorders, they can be used in neuroprosthetics and can help people experience tactile sensations and feedback. This is useful for neuroprosthetic limbs as it makes them easier to use and like more realistic, I guess. And more advanced algorithms and machine learnings are still needed to make this better. So some more clinical applications, they can help with diagnosis. I kind of mentioned that already, but they can also be used to give patients more customized treatments and can predict patients' responses to different treatments before administering the treatments. And for depression, they can deliver using transcranial magnetic stimulation and neurofeedback. They can modulate brain areas responsible for mood regulation and can help reduce symptoms. And this is especially useful alongside deep brain stimulation for treatment-resistant depression. And for OCD, for example, they can be used to identify specific neural circuits associated with compulsions. And for epilepsy, they can monitor brain activity and predict seizures. And they can be used to intervene, and this is especially useful for drug-resistant epilepsy. So overall, some advantages are they can help with personalized treatment and with neural circuit reconstruction. They can provide feedback, which can aid treatment as well and help enhance neuroplasticity. And they can use brain activity as an input signal to stimulate things like both the person's muscles or outside devices. And some drawbacks are if the BCI uses signals from an EEG, EEGs are susceptible to noise interference, so the signals aren't always super clear. And also factors such as surgical and infection risks, and battery storage, portability and comfort, as well as like possible invasive surgeries might make people not want to use BCIs. So clinical efficacy can be tested through assessments of patient recovery. An example of this is the Fugel-Meyer assessment, which is used to measure motor function recovery in stroke patients. And it was found that BCI training can lead to enhancements in patients' FMA score. And this gives us valuable insight into like how BCIs can aid recovery. There's not really standardized measures of rehabilitation for different BCI systems, so it's kind of hard to measure this. But user satisfaction and usability are important points, so if BCIs are more user-friendly, then generally they're more likely to produce positive results. Some ethical implications of BCIs are for user safety. Inserting the electrodes into someone's brain can cause possible injury, and even non-invasive BCIs can have negative effects on neuroplasticity. There's also concerns over autonomy, since we don't really know if an action achieved with a BCI is actually what the user wanted to do, or if it was just like how the brain signals were interpreted, and concerns over how BCIs can affect one's personality. And there's also privacy and security concerns, since like lots of brain data is going to be stored with BCIs, and also stigma and discrimination, since if people use BCIs very conspicuously, they could possibly be discriminated against. Some more ethical implications are informed consent, since some people, such as people with locked-in syndrome, can't consent to BCI use, but we still need to find a way to respect their rights through the BCI use. And responsibility, since we don't know the extent to which the BCI can accurately represent one's brain states, so we don't know if a BCI user should be responsible for all actions they commit using the BCI, as well as who is responsible for if it malfunctions, and how should the law treat brain data. These haven't really been figured out yet. And also equitable access, since BCIs might not be accessible to everyone due to high prices or insurance companies not covering them, so this could further social inequities.
So now we're going to move on to some neuro AI concepts. Last time I talked about closed loop systems within BCIs, digital twins, digital prescriptions. So we talked a lot about how they work, but we're going to kind of dive a little bit deeper into how a closed loop system actually decodes that information. So neural recordings are very, very vast in terms of the data, because we have thousands, if not millions, of neurons within our brain that are constantly moving and firing. And neuroplasticity is constantly happening. Our brain is constantly working. So when you are trying to get that data out of the brain, there is, as Ron said, there is bound to be noise within that data. So research has actually found that even though there's a lot of noise within the data, you can get the same results if you just take a few particular dimensions. I'll get into that a little bit more later within that data. So what we're going to think about is each singular neuron as one-dimensional within a brain state space. And if we're measuring something like from the motor cortex, we have around 50,000 neurons within the motor cortex alone. Now, if we're going to take that data, that would be 50,000 dimensions of data that would have to be processed. So this is a big problem because of the fact that brain data is constantly continuous, and we need that continuous data flow for proper reactions from closed loop BCIs. So that brings neuroscientists to a concept called the manifold hypothesis. So the manifold hypothesis in neural systems is the high-dimensional neural activity is constrained to a low-dimensional space, otherwise known as a manifold, embodied in the larger space. So this can be, as I said before, neurons when they're individually kind of mapped, they are referring to one-dimensional space. So again, like 50,000 neurons would be 50,000 in dimensional space. Neural dynamics for a particular part of the brain is actually only around like 5 to 20 dimensions within a specific state. So the goal to decode the neural data is actually to identify the latent variables within the data that we're representing through the brain state. So the brain activity is not static. I've said this before, it's always changing. And so examples of this could be motor control. We're constantly planning what we're going to do with our hand, and then we're actually doing it, and then based on that we're getting the feedback, whether or not the brain properly confirmed those actions. In terms of decision making, neural activity is moving towards a decision attractor state. This is basically where you're mapping neural activity on a n-dimensional graph. You're plotting those points, and then you're trying to find a stable point within that data for which the brain is trying to get to. So in Parkinson's disease, the pathological oscillations kind of want to be in a stable ground, but in Parkinson's disease, that stable ground is actually not a great outcome for brain patients. In epilepsy, we see that those oscillations and those neural patterns are kind of going towards something called a seizure attractor. So that is basically saying that the stable point within that data is when a patient is having a seizure. So closed loop BCIs do decode all of this, and they kind of determine what the current position on that specific manifold is. And you can think of the manifold as an algebraic topology graph of a specific part of the brain. So for decoding, in terms of the manifold, closed loop systems are able to decode where that particular brain state is on the manifold and determine where that particular state is trying to head to next. It could be towards a healthy part of the brain, it could be towards a pathological part of the brain, but obviously when we're looking at patient data and clinical outcomes, we want those states to go towards the healthy outcomes. So what happens in this situation is they actually trigger either neuron firings towards a better state in the manifold, or they try to kind of create more oscillations that will kind of point that state towards the healthy region instead of the pathological region. So this can be used in adaptive DBS for Parkinson's using something called a Utah array electrode. It's where they have 100 channels that are able to decode all this information in less than 5 milliseconds. Depression, we can adjust the stimulation based off of the decoding that is getting drawn out from this manifold interpretation, and then obviously for seizure controls, we can detect when a manifold is heading towards that seizure attractive state and kind of pull that state out from actually getting to the attractor per se. So this kind of leaves us with the question of last time we talked about digital brains, and now with using this new information, we can say that manifolds are actually, when you're creating a duplicate of your brain, you're actually creating a manifold duplicate of your brain rather than individual neurons from your brain. So the question then becomes, how do you think it's ethical, first of all, and how, let me just read the question. I can't read anything, but this is the question that we can talk about. But yeah, it's a pretty interesting concept. That's the last slide I have.